It's like the 8.
How many traits does a dihybrid cross examine?
4.
So it looks at 2 traits, right? Di, 2. But we look at 2 traits at the same time. So when we do 2 traits at the same time, we have 2 different genes that we're looking at. Because remember, 1 gene is 1 trait. So 1 gene is 1 trait. When we write a genotype for 1 gene, we do both alleles, right? What you got from mom and what you got from dad. So here we're going to look at, let's say, tall and short. And let's go with wrinkled or round. So in pea plants, you're either tall or it's short. The pod is wrinkled or it's a round, smooth pod. That's 2 different traits, right? Independent of each other. They have nothing to do with each other. They just both occur in the same plant. Tall versus short. Tall is dominant, so what are my potential genotypes for the gene tall?
2 capital T's or capital T lowercase t.
And if they're going to be short, round is dominant. So what are the 2 genotypes for a round?
You do 2 capital R's and a capital R and lowercase r. A baby.
And then for the wrinkled,
2 baby
now most of you, if I asked you, actually all of you based on, well, yeah, all of you based on your quizzes, that first Punnett square, no problem. You can do a single trait. You're like, yeah, an individual trait, I'll do those all day happily. And that's when you get back to our incomplete dominance and codominance. It'll be a single trait, 4 squares. You'll happily do them, no problem. It's when we look at the same time that there's an issue. So we're going to look at, two parents, right? Parent 1, parent 2. How many, if they're going to produce gametes, so they're going to make eggs or sperm, in this case it's going to make pollen, will it have 1 gene or both genes in that gamete? Okay. So if it's going to have both genes in that gamete, both the round, wrinkled, tall, short gene, then how many letters will every gamete have? 2. Every gamete gets 2 letters. So let's look at the 4 potential gametes that parent number 1 will make. And just so you make connections from last chapter, it makes them through the process of meiosis. So this would be the result of meiosis. The 4 potential gametes here. First thing it could be is a capital R, right? Now it's going to pair with an allele from the second trait. So what are my 2 choices for the second trait? Capital T or lowercase t. So the first one, it's going to pair with a capital T. Capital R could also pair with a lowercase t. Moving on to lowercase r. These 4 gametes are what I'm going to transfer to the sides, right? So go ahead and make that transfer in your notebooks if you need to. So we agreed it'd be capital R, capital T, capital R, lowercase r, capital T, lowercase r, lowercase t. Same thing across the top because it's our other parent. Go ahead and fill in that Punnett square now. Like terms collected. So don't go, for example, down here, don't say capital R, capital T, lowercase t. You have to collect like terms. So R gets paired with the other R, T is paired with the other T. Usually this is in alphabetical order. Not always, but quite typically. Those of you, well, we'll stop and say this. Any questions about that? Make the gametes for this one.
Wait, are you telling us today what we got on our chest? Are you
Hopefully
gonna,
at the
like,
end,
show
I'm gonna,
us?
hopefully at the end I'll have a moment to you. I just wanted to make sure I had full and complete attention. Don't have to do a Punnett square for it, just show me what my 4 gametes are. What's the first gamete?
RT. Capital R baby T.
And the second gamete matches that, right?
Yeah.
And the last 2 gametes are?
Baby R, baby T.
Make the gametes for this particular individual. What's the only possibility for an allele for the first trait? The only possibility is capital R. And how about really? 1? What? So she's O. What other choices are there? So we've heard positive and negative, right? When we talk about blood types, that positive or negative is actually, that would be like looking at 2 alleles at once. So if we looked at both blood type and the positive negative thing, we would be doing a 16 square Punnett square. We're going to start with just the letter. So that way we can start with Punnett squares of 4. Better? It turns out that you only have 2 alleles for blood type. One you got from mom and one you got from dad. Just like every other trait you have in question. But unlike everything we've looked at so far, we've only seen 2 potential alleles. So for example, tall and short, your choices are a tall allele and a short allele, a capital T or a lowercase t. and there are no other options. There are genes that exist that have lots of other options, and blood type is an example of it. The potential alleles that you can get are one that makes for O blood, one that makes for A blood, one that makes for B. But this isn't classic Mendelian inheritance because they're not dominant over another. So we're going to make a chart of your blood type. So we said O, A, B, AB. Those are our choices. So your blood cells look like Smarties. They are shaped like that. They have a dent in the middle of them. But on the outside, their surfaces aren't always smooth. Sometimes there are proteins sticking out of the surface, and each of those proteins has a different shape depending on the protein it is. Type O blood does not have any proteins on the surface of it. So it's just a Smartie-shaped cell with nothing sticking out off of it. Type A blood has that same Smartie shape, but instead there are proteins on the surface that have one particular shape. Type B blood, again, the same Smartie shape, but it has a different shaped protein sticking off the surface. That's why you can't put type A blood into a person with type B blood, because the shapes of the proteins on the surface indicate that it's a foreign invader, so it starts to attack. That not only is it not good, it will kill you. So we try and avoid that. It's usually bad. People with type AB blood have both shapes on their cell surface. So it's a combination of the A and B. And the reason for that is because they have one allele that codes for the A shape and one allele that codes for the B shape. People with type O blood don't have any. So the allele that they get, the genotype, we call it lowercase I. Lowercase I, it's lowercase because it's recessive. When it's present, it's always masked. So if that allele is present, you never see it unless you have two of that allele. Somebody with type A blood, cell. If you're type O blood, you have nothing on the surface of your cell, which means your blood can be put into anybody else, and their body won't react. There's nothing on the surface to indicate that it's a foreign invader. If you're type AB, your body will accept type O blood, because there's nothing to indicate it's an invader, type A blood, because your blood also has type A on the surface, or type B blood, because you have type B. So if you're type AB, you are called the universal recipient, because you can get blood from anyone else. But if you're type O, you're the universal donor. If you're type A or you're type B, you can only give to other type A's, and you can only accept from type A. So that's the difference there. It is not the most, I only know the breakdowns by race. I don't know generally speaking. It is not the most common of the blood types. I know that. I think it's O. So let's see this way. Take a mother who is heterozygous for type A. What's heterozygous for type A? What's her genotype going to be? And a father who is type O. Here you have a simple Punnett square again, right? We get to return to the happy squares of 4. Cross these two.